Welcome to video 2 in our lesson on cardiac muscle. In this video we're going to talk about the process of excitation contraction coupling. The excitation part more than the contraction part. We'll cover the contraction part in the third video. So as you guys know, action potentials propagate from cardiac cell to cardiac cell, and they do that along the surface membrane here, which is called the sarcolemma in muscle, in striated muscle. And the action potentials propagate from one end of the cell to the other, and as they do it along the surface membrane, they dip down into structures which are called T-tubules. And these T-tubules are a chronic source of confusion for medical students because they have a tendency to think of them as organelles, which you probably should not do. These T-tubules are nothing more than invaginations of surface membrane. So think about this, the membrane coming here, and the membrane just dips down into the middle of the cell and then comes back up and then continues on. But the T-tubule membrane is continuous with the surface membrane. It really is surface membrane. It's just surface membrane that's dipping into the cell and coming back out again. Okay? When we talk about the inside of a T-tubule, that's basically extracellular space. You know, this is still cytosol here, and this is basically extracellular space. You can see there's this opening here, and the extracellular fluid is continuous with what's in the T-tubule, and the components are basically the same. So don't think of this as some sort of a compartmentalized organelle. Think of it as just an extension of the cell membrane down into the middle of the cell, okay? Containing extracellular space, and as your action potential dips down, all you're really doing is you're bringing the electrical signal down into the middle of the cell, okay? Here is our cardiac action potential, which I introduced you to in the introductory lesson to this block. The resting potential about minus 90 millivolts. And you will see the cardiac action potential is compared to the skeletal muscle action potential. And you can see right away that the cardiac muscle action potential is much longer than the skeletal muscle action potential. And the reason is because of the existence of voltage-gated calcium channels in the membrane of the cardiac muscle. So what's happening here is, these are called L-type calcium channels. And what's happening here is the membrane voltage is going up, and as that membrane voltage goes up, these calcium channels open, okay? And they allow calcium to flow into the cell. Well, when you allow calcium to flow into the cell, what that does is it brings positive charge in that you ordinarily wouldn't have, say, in a skeletal muscle. So the skeletal muscle action potential comes right back down. But because you've got this extra positive charge coming in, making the inside of the cell more positive relative to the outside of the cell, the membrane voltage hangs up instead of coming right back down in the cardiac action. And the cardiac action potential, it hangs up. And you get this prolonged action potential that's more like about 250 milliseconds long rather than about 2 milliseconds long. Okay? And that's because calcium is coming into the cell, and that's because the calcium is extremely important for causing cardiac muscle to contract. Okay? Cardiac muscle cannot be tetanized. What we mean by tetany is that if you take a skeletal muscle cell and you just hit one action potential after another, bang, 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 bang. What happens is your skeletal muscle cell contracts, and it contracts more and more and more and more as you add one action potential after another in that cell membrane. So that does not happen in cardiac muscle. And the reason it doesn't happen is because you can't throw another cardiac muscle action potential until the first one is done. So the voltage has to return back to the resting potential or near the resting potential before you can stimulate this membrane to throw another action potential. Well, you can see at the end of this cardiac action potential, cardiac muscle is almost all the way relaxed. So by the time you can throw another action potential, the cardiac muscle is almost back down where it belongs. So because of that, you can't just fire one action potential after another and cause the force of contraction to build. In skeletal muscle, you're throwing one action potential after another, and you're not allowing that muscle to relax. In the case of cardiac muscle, it does relax most of the way before you can throw another action potential. So we say that cardiac muscle can't be tetanized, skeletal muscle can. And this is not a bad thing. There are no circumstances under which you would want your heart to contract and not be able to relax. If you can't relax, you can't fill. If you can't fill, you can't eject blood. If you can't eject blood, you die. Okay? So this is not a bad thing. It's actually almost a safety factor. Okay? All right. So the other structural element that we want to point out in cardiac muscle is the sarcoplasmic reticulum, or the SR, here and here. The SR is an intracellular organelle. Unlike T-tubules, they are actually membrane-bound, they are in the cell, and they are a compartment in the cell, a compartment which is separated from the cytosol by a membrane, the SR membrane, or the sarcoplasmic reticulum membrane. Okay? These membranes contain a very high density of calcium pumps, that is, SR calcium ATPases. They use ATP to pump calcium from the cytosol into the sarcoplasmic reticulum, where it is stored, and from which it is released during contraction. Okay. The SR has what are called lateral sacs. You know, this is all SR in here, and this is all SR. But the lateral sacs are sections of SR that come very close to the T-tubule, here and here, and here and here. Okay, and because they come very close to the T-tubule, they're sort of almost in communication with the T-tubule. And the fact that they're very close makes it easier for this action potential, which is propagating down the T-tubule, to send its signal to the SR to tell the SR to release calcium. Okay, so these are areas of the SR which come in very close very close to the sarcolemma. They don't connect to it, but they're very close. Okay. So the region in which the T-tubule membrane comes in close proximity to the SR, where these lateral sacs are, is called the T-SR junction. And a lot of the action in excitation, contraction, coupling takes place there. Okay? The SR membrane, where the lateral sacs are, contains ryanodine receptors. These are otherwise known as SR calcium release channels. So as you might imagine, calcium is going to be released from the sarcoplasmic reticulum through these channels into the cytosol to cause contraction of cardiac muscle. Okay? So let's talk a little bit about how that takes place. Okay? Now that we've seen the players, we have our action potential. It's propagating along the sarcolemma, which is the cell membrane. Okay? And as it propagates, it opens up channels in the cell membrane, which are the L-type calcium channels. The increase in voltage opens those channels, and calcium flows in through those channels. Okay? The action potential also dips down into the T-tubule, and this is a lateral sac here in your SR. So, calcium comes in, there you go. And these calcium channels are opening up in this T-SR junction. And when they open up, that calcium that comes in through these L-type calcium channels binds to the ryanodine receptors, or the SR calcium release channels, and it causes those channels to open. So what we get is what's called calcium-induced calcium release. That is, calcium coming across the T-tubule membrane, across the sarcolemma, through the L-type calcium channels, binds to the ryanodine receptors. The ryanodine receptors open, and calcium is released from the sarcoplasmic reticulum into the cytosol. Calcium is high in the SR, lower in the cytosol. It goes from high to low through the channel, and calcium increases further in the cytosol. Okay, so the calcium that's coming in through the L-type calcium channels combined with the calcium that's being released from the SR causes a rise in cytosolic calcium. About 20% of it comes from the L-type calcium channels, about 80% from the sarcoplasmic reticulum. Okay? And this is what's going to cause your myofilaments to contract and generate force in the muscle. Okay? And what I want you to notice is that, we made this point during the host defense block. Calcium does not have to diffuse very far because of the existence of T-tubules and the SR. The reason why you have T-tubules is to take this action potential and make it propagate down into the interior of the cell so that it can be where the myofilaments are. The myofilaments are running here through the cell. And so what you're doing is you're causing the action potential to fire really in the interior of the cell, and that's going to bring this calcium in through the L-type calcium channels deep inside the cell, close to the myofilaments, so it doesn't have to diffuse. If it had to diffuse from here, from the cell membrane, the surface membrane, it would take a long time. But if you can bring that calcium to the myofilaments, then it doesn't have to diffuse very far. And you know that when you're using diffusion to have a substance, to transport a substance, the distance of diffusion has to be very small. And the T-tubule keeps that distance small. The fact that the sarcoplasmic reticulum is very close to the myofilaments and wraps around the myofilaments means that that calcium never really has to diffuse very far in order to be released, or as we're going to talk about in the next video, taken back up into the sarcoplasmic reticulum and reaccumulated for the next beat. Okay? And here is a summary of our excitation protection coupling. The end plate potential you can forget about. That shouldn't be here. That's only relevant to skeletal muscle, but this is relevant to cardiac muscle. You have action potential propagating down the sarcolemma, and that causes the T-system action potential, and that brings calcium into the cell, and calcium is released from the SR via calcium-induced calcium release. Okay? Forget about that, but this is relevant. This is basically excitation in a nutshell. Okay? All right. That's all I've got for this video. In the next video, we will continue on, and I will talk about the way that the myofilaments contract in response to calcium, and we'll talk about the process of relaxation briefly after that. Thank you.